Okay, so I want to go over this paper, which is a short segment of prion protein plays a critical role in its susceptibility to cross-species prion transmission. And it's not really a paper. The paper is actually down here. So here's the DOI if you want to go to it. So this is in the Nature Chemical Biology. So it's short disordered protein segment regulates cross-species transmission of a yeast prion. So what are prions explicitly? So prions, these are proteins that are misfolded, or we like to say that they're misfolded, but granted, there theoretically could be some evolutionary trait to have them fold in that particular method. However, for in the context of diseases, we call them misfolded proteins. And so basically, these proteins, especially proteins that are in high concentrations in certain parts of the body, these proteins might misfold and then crystallize or form polymeric structures between them. So I think we'll just go to the original paper and continue. So a short segment of an infectious protein known as a prion protein plays a crucial role in determining how susceptible the protein is to interspecies prion transmission. So remember, these prions, they're proteins, and proteins are made out of amino acids. So when you know, eat your protein powder, they'll leave the amino acid composition in it so that you can keep track of the amino acids you're eating. So here we have a short segment, so a short amino acid sequence that is a part of a larger protein. So the larger protein itself can form these prions, but only a small amount of the protein is actually a part of the prion producing process. So in determining, and let's see, so granted, those are prions that are in, how to phrase that? So you can have prions that are within one species, but we want to look for different types of sequences that can determine whether or not a prion won't just stay within the same species, but will be able to make the jump into another species if they try to consume the material from the first species. So what to me would indicate this? Well, this would mean that whatever sequence that is, that sequence probably needs to be conserved or more conserved in multiple species, right? So if you have a highly variable sequence that is doing this, then one species versus another might not have that same sequence, and therefore it might end up that they can eat the prion in one species, but then that prion won't be able to misfold the proteins of that other new species that is consuming it. So here, RIKEN researchers have discovered in a yeast study. So yeast, these prion proteins, you can, let's see, let me go to this and see if this is a natural part. So here, the soluble prion proteins contingently encounter foreign prion aggregates. So remember, these prions, they form aggregates, and soluble prion proteins will eventually, as they're floating around, pop into or meet one of these aggregates. And then this could cause cross-species prion transmission. However, how its efficiency is regulated by structural fluctuations of the host soluble prion protein remains unsolved. So this is what is basically happening. In the host, they can have these proteins that are possibly going to be prion forming. They are primed, or they're ready to form a prion. But if there is no aggregate, they will never form, or I should say it'll be very difficult for those soluble prion proteins to actually form an aggregate. So once an aggregate gets into the body of this poor creature, then the soluble prions might be able to interact with that aggregate and then misfold themselves, and then they can grow like a crystal structure. So you could imagine this like the nucleation of, I don't know if you've seen videos of people when they boil water or bring water way above its boiling point, but the water is still maintained at that as in the liquid state, and it takes a nucleation site, or it takes something to disrupt that water to basically have it all explode at once. That would be an example of what is happening here, where the prions could theoretically form or come into these aggregates, but they need a nucleation site first. They need something to attach to break that energy barrier. Anyways, in the present study, through the use of two distantly related yeast prion SUP35 proteins. So remember, these prion proteins, or these I'll just say these prion proteins, or these proteins that could very easily turn into prion diseases. These proteins can be seen all over eukaryotes, and I would assume possibly some bacteria might also produce proteins that are similar. However, these yeast proteins, which are eukaryotes, are producing these proteins that can theoretically form aggregates as well. Anyways, they took two distantly related yeast proteins, and we found that a specific conformation of a short disordered segment governs interspecies prion transmissibility. So these proteins that are likely to form these structures, they will have small parts of their amino acid sequence that are not structured, or they are disordered normally, and that's how the protein normally wants it to be. So proteins will often have a secondary structure. And the secondary structure could be the alpha helices, where the amino acid sequence forms a helix. You could have beta sheets, where the amino acid sequence will line up straight, and then it will bond with another straight beta sheet or another line of amino acid sequences. And then if you have multiples of these sheets, you can form planes. Regardless, those would be ordered versions of amino acid sequences that, while they order, they actually release energy and are stabilized. But there are some purposefully unstructured or what would the word be? Disordered sequences that are in proteins that are necessary for their function. But since they're disordered, they might have the possibility to order in a specific way. And if these disordered segments end up ordering themselves with the disordered segments of a different protein, then you can start getting these aggregates, right? So this is how they could cause the nucleation or the precipitation of the soluble form. So let's continue. So using a multidisciplinary approach, including the high resolution NMR, which this is just basically it's an MRI for a small solution or a small bit of a solution. And when you do this MRI on the small bit, you can actually get structural information. So they did high resolution NMR and molecular dynamics simulations, and we identified critical residues within this segment so that allow interspecies prion transmission in vitro and in vivo. And we did this by locally altering dynamics and conformation of soluble prion proteins. Remarkably, subtle conformational differences caused by a methylene group between asparagine and glutamine suffice to change the short segment structure and substantially modulate the cross-seeding activity. So if we just add this small methylene group, which would be a carbon with a few hydrogens, if we add that in between the asparagine and the glutamine, which are two different amino acids, they can substantially modulate this cross-seeding activity. Thus, our findings uncover how conformational dynamics of the short segment in the host prion protein impacts cross-species prion transmission. More broadly, our study provides mechanistic insights into the cross-seeding between heterologous proteins. So these are heterologous, meaning they are completely different proteins. So here, remember, they are talking about two different yeast proteins, and somehow one of these foreign SUP35 proteins was able to cause the precipitation or the prion formation of this different soluble protein, right? So this is what they're calling the SUP35s, and I don't know exactly what it's used for, but just know if you have these prions in one yeast, they can get into another yeast and cause this seeding, or they could be causing this nucleation site for these soluble proteins to fall out of solution. And apparently, the only part, or the most important part, isn't the entire protein, but rather it is just a small part of the protein, a small amino acid sequence that is doing this. So let's go back to this article. So the infectious diseases are usually spread by living pathogens such as fungi and bacteria, or by genetic material encased in protein shells such as viruses, which is pertinent right now. But proteins that are identical to functional ones found in the brain but misfold into aggregates, this misfolding can cause fatal infections in people by inducing correctly folded proteins to switch configurations into an abnormal form. So if you say were to eat a cow that had mad cow disease, or I think there is a sheep version of that as well, or if you eat um, if you're cannibalistic, that can happen as well. But basically, you can have these misfolded protein diseases moving on or passing on into the host or the new host, right? And so basically, and actually, what's really interesting about that is proteins are usually degraded by the digestive system, right? So your body breaks down these proteins into their constituent amino acids. Now, however, this process might not be completely perfect, and you might still have small peptides left. So this would be an indicator that at least to me, that the sequence that can start causing these aggregates could theoretically just be a very small amino acid sequence, and that would be one of the ways that it could theoretically get through your system and then cause disease elsewhere. So anyways, while relatively rare, such prion infections have no cure and are always fatal. So don't get it, we don't have a cure yet. Oh, remarkably, such prion infections can happen between different species. The best known example of this observation is a variant of this Creutzfeldt-Jakob or Jacob-Jacob disease. I don't know how to pronounce it, or CJD, and it can be caused by eating meat from cows with mad cow disease. So scientists would like to understand more about what properties of the monomeric prion protein increase its ability to cross species. And granted, I do not like how this article is being written. It seems very run-on sentencey to me. However, but it's challenging to investigate because the transmission efficiency is quite low. If, say, the cross-species transmission efficiency is less than 0.1%, you would need between 1,000 and 10,000 mice to get meaningful statistics for just one experiment, which is almost impossible. So we can say that this is probably a very good thing, right? It's good that these prion diseases don't readily or don't easily cross between species, and I would presume that that's likely because enough amino acid sequences change in between species that it'd be very difficult to have that proper amino acid sequence one misfold into a prion or a prion plaque, and then to have a closely related species eat that other one and just so happen to have a large enough chunk of it get through the system in order to progress the disease further. So yeah, very, very unlikely, and it is good that that is unlikely. So anyways, to overcome this problem, Tanaka, these people have turned to prions that infect yeast. In yeast systems, we can easily screen prions containing species barriers in thousands of colonies in just one day, and that is a huge advantage. So rather than having to deal with trying to infect animals, let's just deal with yeast, because with the yeast, you can look into each of them, you can see whether or not the aggregates are forming, and you can do a lot of mutational studies, right? So you can mutate yeast, and there's not going to be ethical or nearly as much ethical considerations that they have to take into account. So yeah, do the work on the yeast rather than do the work on the animals. So by using two distantly related yeast prion proteins from different yeast species, the team discovered that a change in the shape of a short segment of a monomeric protein can substantially affect how easily prions can jump species. Remarkably, the difference of only one methylene group in this region of the monomeric yeast prion protein can alter its dynamics and hence how easily it can react with prions from different species. This came as a very big surprise to us. As a result, a fresh perspective for studying human neurodegenerative diseases such as Alzheimer's and Parkinson's disease. And remember, Alzheimer's is very similar to a prion disease, where you have these amyloid beta plaques that are building up. These amyloid beta plaques, they're basically misfolded protein that will cause more of the soluble protein to misfold and then grow the aggregate. And granted, this is happening in the extracellular space or outside of the cell, but as they grow, they can actually damage the nearby nerve cells and therefore cause Alzheimer's disease. So many researchers have been focusing on amyloid structures because amyloids can cause functional monomeric proteins to become dysfunctional by recruiting them into aggregates. And our study provides a novel viewpoint, namely that dynamic fluctuations in the structure of monomeric proteins is also critical. So stepping back, this is what I think that this is very useful information. And for one, I think what would also be very interesting is to do these transfections of these yeast with, say, human amyloid beta proteins. And granted, you wouldn't need the whole protein; it's only a small portion of the protein is called amyloid precursor protein, or APP, and only a short segment of it is actually causing these amyloid beta plaque buildups. What would be interesting would be to just take that short segment, or that shorter segment that is known to form these plaques, and transfect them into yeast and see, by doing mutational studies, what parts of the amyloid beta, or what parts of the amyloid beta amino acid sequence, or the peptide, what parts of it are important in forming these plaques. And by doing that, we might possibly learn different types of genetic engineering techniques that might theoretically help prevent Alzheimer's. We might think of different types of, what would the word be? Different types of peptide drugs could theoretically be given that might be able to help break up these peptides by interfering with those amino acids coming together. So yeah, I think that there's a lot of potential in this mode of studying by using yeast as a model organism rather than having to uh, deal with people, right, or deal with animals. Granted, when we're talking about amyloid beta proteins, we're talking about an in species, or something that happens within the same organism, right? And I guess the original purpose of this research was to look in between species what amino acid sequences are important for this interspecies prion diseases. But if you learn about that, you are also going to learn about these aggregates even within yourself. So science isn't just one order of effects; it is a multi-ordered or a multi-ordered effect on society. So with that, I'm going to head on out, and I will leave the citations for this and the paper in the description if you want to read it for yourself. And hopefully, I will see you tomorrow.